When last we spoke, we dealt with the problem about how to keep glucose from leaving the cell. For those that don't remember the previous video, we remember that the overwhelming majority of the cell membrane structure is made out of phospholipids, and since the interior of the phospholipids is nonpolar, that means that glucose cannot cross the cell membrane without the aid of a carrier protein. And of particular note, to help us for our strategy to keep glucose inside of the cell, we remember that the outsides, both the outside layer facing outside of the cell and the outside layer facing inside the cell, obviously have a phosphate, which, as we know, has a strong negative charge. What does this have to do with our strategy to help glucose stay within the cell? Well, as a hint, the way that carrier proteins work is that they have an active site that has complementarity, both in terms of size and polarity, that restricts only a specific substance from being able to get into the cell, but not other substances that have a different size and a different charge. Therefore, if glucose's structure was altered in some way, we would expect that it would not be able to fit inside of the carrier protein anymore, which means once glucose enters the cell, if its structure is altered in some way, it would not be able to get out using the same carrier protein, even if the concentration gradient made a higher concentration of glucose inside the cell than outside the cell, supported glucose diffusing out. So what can we do in order to change glucose's structure? Well, we remember that if the outside layer of the phospholipid bilayer is negatively charged because of phosphate, we can also add a negative charge to glucose by literally putting a phosphate on glucose, and that's exactly what we see. So this molecule here, we can see that there is a phosphate that is now attached to glucose, and we can see that that phosphate is attached to carbon 1, 2, 3, 4, 5, 6. Hence why we refer to this molecule as glucose 6-phosphate. So, where did this phosphate come from, and how did we get it onto glucose? Well, as we know, most phosphate is transferred between different molecules within the body or within a cell using ATP. And if you remember the mechanism that we use for nucleotide polymeration, this mechanism works in a very similar way. So using an enzyme that we call glucose 6 kinase, we can catalyze a reaction involving glucose and an ATP molecule. So a kinase is a family of enzymes, and their job is to add phosphates onto specific molecules, and not only does it tell you which molecule to which we're adding a phosphate, but even the correct location, hence carbon 6. So the way that glucose 6 kinase is going to work is that its job is to pull this hydrogen off of carbon 6, resulting in this bond breaking and the electrons in this bond becoming a lone pair on this glucose. Now, as a consequence, one of these lone pairs is naturally going to move to an ATP molecule that is located in the active site and make a bond with the phosphorus atom in the first phosphate, breaking this bond here, and now successfully attaching our phosphate molecule onto carbon 6 of glucose. Now, as a consequence of this, glucose is now no longer able to fit into the active site of the carrier protein, nor does it even want to be close to the surface of the cell membrane, which means that this glucose molecule now no longer can exit the cell. But there is an additional benefit because we have made this glucose molecule less stable as a consequence of this negative charge. Why is this? Well, remember that every single oxygen in a glucose molecule, because it has two lone pairs and a high electronegativity, is going to have a partial negative charge on it like this, including the oxygen that it attached to right here. Now, we know that ATP is unstable because of all sorts of intramolecular repulsion between the negative charges on the phosphates, and the same principle applies to glucose, even though the repulsion is between the full negative charges of the phosphate and the partial negative charges of the individual oxygen poles. Now, unfortunately, this means that glucose with a phosphate, so glucose 6-phosphate, is not unstable enough to spontaneously break down. But if we make glucose 6-phosphate unstable by adding a phosphate, logically what we can do is simply add another phosphate ion in order to make glucose even less stable and more able to break down. But first, we are going to make a change. So here we see glucose 6-phosphate, and this molecule here is a compound that we call fructose 6-phosphate. And fructose is simply an isomer of glucose, meaning that it has the same chemical formula, C6H12O6, including the phosphate that is still in the same location, but we see that its structure is different. And the enzyme that is going to catalyze this particular change is an enzyme that we call glucose 6-phosphate, referring to the target molecule, and its job is to make an isomer, so we call this enzyme an isomerase enzyme, meaning it's going to change the structure of glucose 6-phosphate without changing the chemical formula. What is the point in doing this? Well, the next phosphate that we are going to put onto fructose 6-phosphate is actually going to fall on carbon number 1. Now, as long as we have an alpha glucose molecule here, we can see that carbon 6 and carbon 1 are pointing in opposite directions like this. But here we can see that carbon 1 and carbon 6 are pointing in the same direction, which means that the two phosphate molecules that would be added to fructose 6-phosphate are on the same side of the molecule, making the repulsion force between them even stronger and making this molecule even less stable than the glucose form of it. And that's exactly what the purpose of this isomerization reaction is. So once we have a fructose 6-phosphate, the next enzyme, which has the rather clunky name phosphofructo, let's get the spelling correctly, fructokinase, which we can simply abbreviate as PFK, phosphofructokinase, is going to do the same thing as glucose 6 kinase did. Our enzyme is going to pull this hydrogen off of the OH group on carbon 1, turn this bond into a lone pair, and then one of these electrons is going to bind to the phosphorus of the first phosphate of a second ATP molecule in order to make this phosphate here on an ATP molecule attached to carbon 1 of the new fructose molecule, giving us a new molecule which we will call fructose 1,6-bis, bi of course being the prefix for 2, bisphosphate, because of course we have two phosphate molecules on this structure now. And now that we have done this, this structure is definitely not stable because of the very strong repulsion forces that exist between one phosphate ion on carbon 6 and the other phosphate ion on carbon 1. So naturally, we would expect this structure here to break, and with the help of our next enzyme in this process, an enzyme that we call aldolase, and we'll talk about what the significance of this is, this molecule is basically torn in half, where 1, 2, 3 carbons, or rather, if we label these properly, 4, 5, and 6, this half of the molecule containing carbons 4, 5, and 6 basically becomes this molecule here, which we can call glyceraldehyde 3-phosphate. We can recognize that there is an aldehyde functional group here, meaning a terminal carbon with a double bonded oxygen here, and with the phosphate on carbon 3. And we can simply abbreviate this as G3P. While the right half of this molecule, carbons 1, 2, and 3, become our second molecule here, which is what we call dihydroxyacetone phosphate, also abbreviated as DHAP. So as a result of these intramolecular repulsive forces between the phosphates that make fructose 1,6-bisphosphate unstable, allows the enzyme aldolase to spontaneously tear this molecule in half. And to conclude our first step, we can take dihydroxyacetone phosphate and convert it into a second molecule of glyceraldehyde 3-phosphate, resulting in 2 glyceraldehyde 3-phosphate molecules. And the mechanism of doing this is basically taking this OH group, which we would normally find on carbon 3, and moving it to become an OH group on carbon 2 instead. And the reason behind this will become evident in the second half of the glycolysis process here. So because all that we're doing is flipping the position of these double bonded oxygens and OH groups, we are doing another isomerization reaction, which means that our enzyme is going to be another isomerase, only this target is going to be dihydroxyacetone phosphate isomerase, also simply called triose, meaning 3-carbon sugar, phosphate, meaning that there's a phosphate on it, isomerase. What this means is after we have 2 molecules of glyceraldehyde 3-phosphate, every reaction that we are going to see in the second half of glycolysis is going to happen twice. This concludes the first half of the glycolysis process here, again involving the breakdown of glucose after attaching a phosphate to keep it in the cell. Before you move on to the second half, try a few of the practice problems below in order to understand not just what happens, but why each step that we've talked about here occurs.